Hello everyone, welcome to chapter 16. In this chapter we're going to be talking about disease and epidemiology, and it's very fitting to end the semester with epidemiology because if you look at what's going on in the world, we're dealing with an epidemic right now. The epidemic is COVID, and it is a pandemic, which means that it has spread all across the world. So what is epidemiology? Epidemiology is the study of disease in populations. And I don't know if you remember, but the father of epidemiology and our first epidemiologist was John Snow. His study was on the cholera outbreak in London. He went all across the city trying to find where this disease started, and he was the one who figured out that it was coming from the water well supply, and those who were drinking from that well supply were getting very ill, and when they shut that water down, people got better. And so he is known as the father of epidemiology. What an epidemiologist does, and this is before we had this term epidemiologist, an epidemiologist studies the pattern of disease, the geographical distribution of disease, the occurrence of disease, and they determine how it's transmitted. The goal would be to recognize and control outbreaks. So basically, an epidemiologist is a detective in the microfield. And they design infection control strategies. This allows them to prevent or predict how disease might spread. So there's a lot of terms associated with epidemiology, definitions basically, that you need to know. The first one is called morbidity. The morbidity is the state of being diseased or unhealthy. A morbidity rate then is the number of individuals that have a disease out of a specific number of individuals in a population. So it's a percentage of that population. An example would be if 400 individuals have a disease, and the population is 4,000, the morbidity rate would be then 10%. Mortality is the number of deaths in the population. We can look at prevalence and incidence as well. Prevalence of a disease is the number of cases at a given time. So you can see the prevalence here is this red line of HIV infections. And so you can see in 1980 there were very few. And then it grew to over 200,000, over 400,000, 800,000, and it just kept growing and growing. The incidence is the number of new cases. So it's going to be a smaller number, but the incidence is going to cause the prevalence to go up. So in 1985, we had a high incidence, which means a lot of people were infected in 1985. And now we have very low incidence compared to, well, 1985. A sporadic disease is a disease that is seen only once in a while in a population. And there's no real reason for it. It just pops up. So an example would be tetanus. An endemic disease is one that is always present in a population. It's not always in high levels. It's usually at low levels, but it's always there. So an example of an endemic disease is malaria, which is endemic to regions like Brazil and certain areas of Africa. An epidemic disease is a disease that you find a lot of cases in a very short period of time within a certain region. So an example for this would be flu season or cold season. We usually have epidemics. And then a pandemic is an epidemic that occurs on a worldwide scale. So something that occurs everywhere. COVID-19 is a pandemic. So when an epidemiologist is tasked to determine how to prevent some disease, the first task is to determine what the causative agent is. And if you remember, the causative agent or the etiological agent can be determined by using Koch's postulates, which we talked about in the last chapter and in the beginning of the semester. So in Koch's postulates, it states that the agent, the etiological agent, must be present in every case of a disease. You must be able to grow that agent in a pure culture. You must be able to infect a healthy host, and that healthy host must become diseased. And you must be able to recover the etiological agent again from the newly diseased host and grow it in a pure culture again. So epidemiologists have different types of studies that they do to determine causation of disease as well as how fast a disease might spread. So there are observational studies where they're just looking at a disease and a causative agent and kind of getting information through interviews of individuals that have been infected. Then we have experimental studies where we actually are using Koch's postulates. And this provides a strong evidence for which agent is the etiological agent of a disease. There's descriptive epidemiology. So in descriptive epidemiology, you're going to get information about outbreaks and how disease is spread over time. So this is kind of an exploratory study. There's analytical epidemiology, which can be broken into two different types of studies. So you have retrospective studies, gather data from past on a present day case. So you might take information that you learned about in the past for maybe influenza, and you use it for an outbreak today. Prospective studies follow individuals and monitor the disease state during the course of the study. So now they're looking at individuals that are infected and ill. Apologies, hold on. Now it's jumping all over the place. Okay. So we have certain organizations that help us with epidemiology studies. The Centers of Disease Control is a national organization that does disease surveillance around the entire U.S. and they actually publish morbidity and mortality weekly reports on different diseases. So any diseases, specifically diseases that are uncommon that doctors don't normally see, are going to be reported to the CDC by those physicians and health departments. So now let's look at where these diseases are going to be living. So pathogens have a natural habitat in which they normally reside, and that is known as their reservoir. Reservoirs can be in animals, where things like rabies would be found, can be in humans. And if a human is the only organism that the reservoir can survive in or can reside in, then it's oftentimes easier to control. And also we can have reservoirs be environments. You can't really control an environment as effectively, but things like tetanus would be found more in an environment. So determining where the reservoir is going to help us in controlling those pathogens. Humans, like I said, can be reservoirs. Sometimes we are reservoirs for an infection that only can infect humans, and those are easier to control. But then a lot of times infections can be found in humans, but they can also be found in other environments, and those are harder to control. You can be a passive carrier where you have a pathogen on you, but you are just mechanically transmitting it. So something like you might touch something that has bacteria on it. Say you pick up someone's Kleenex and they have, I don't know, let's just say, oh, why can't I think of it now? Diphtheria. And they were blowing their nose and coughing, and you pick up their Kleenex and you throw it away and you get Corynebacterium on your hand. You can then touch somebody else and transmit it without actually being infected yourself. You can be an active carrier. Active carriers can be symptomatic with an infection. So you are sick and you're passing it on. But you can also be asymptomatic. So I always think of symptomatic infections like a cold. How many people have went to work and they have a cold? Well, colds typically don't keep you home. But colds also get passed on very easily because people are coughing or they have runny nose, and so that can be passed on very easily, and that would be a symptomatic infection from an active carrier. Asymptomatic would be something like a cold sore. So the herpes simplex virus can be asymptomatic, but it can be transmitted to others still. Or HIV is another example of an asymptomatic infection. Sometimes we have healthy carriers. Healthy carriers are very important. If you are asymptomatic, you don't know you have a disease, you can still carry that disease and pass it on. An example of an asymptomatic healthy carrier was Mary Madden. I can't think of her last name now. Everybody called her Typhoid Mary, but she actually was Mary Malone or something. Mary M, that's what I'll call her right now. She was a carrier of typhoid. Typhoid was a serious disease, and a lot of people died from this disease. It's transmitted through fecal matter typically, and it causes severe diarrhea, dysentery. Mary Mallon, that's what it was, Mallon. Mary Mallon was a healthy carrier, so she had typhoid, but she was not ill. And she was a cook. So if she was cooking food, she could pass the bacteria onto her the people that she worked for if she wasn't actually like cooking it, if she wasn't actually heating it up, baking it. So like she would make ice cream that was really yummy, but you don't bake ice cream, and this ice cream actually caused a lot of disease because she was spreading the Salmonella typhi. So it was really bad. There's a lot of videos, or really cool videos that you can watch on Mary Mallon. She was actually not a bad person, but I mean, when you have a disease like typhoid and you're not sick, you still have to be thoughtful of others. It would be like having HIV. You're not showing symptoms, but you still are thoughtful of others. You would never just willy-nilly have intercourse with somebody if you have HIV, because you know better, because you can pass it on. You might not be actually ill, but you can still pass that on. So you take precautions. Well, she couldn't or didn't take the precautions she should have, and because of that, she ended up causing a lot of deaths by accident. Very interesting story, though. Non-human reservoirs. These are going to be zoonotic diseases. So animal reservoirs, zoonotic. They can be transmitted to humans, and a lot of times these diseases are really serious, like plague, rabies, very serious. Then you have environmental reservoirs. So if it's a zoonotic disease, you can still treat by knowing that reservoir. You can lower the amount of the animal in the environment. So like plague was the rat was the common organism that was the reservoir, and then the rat fleas would bite and they were vectoring this disease. So the fleas were not the reservoir. Fleas were actually dying as well. They were just as well, yeah, they got sick and died just like humans did. But controlling the rat population, we were able to lower the number of deaths. Environmental reservoirs you can't really get rid of because, I mean, you can't get rid of the environment. I mean, not in general. So like tetanus, botulism, those are going to be around forever because they live in the soil. Diseases can be transmitted in multiple ways. Hold on one second. I'm going to stop here and I'll put the last few slides in a shorter video, okay? All right, bye.